I'm Gail. I'm the clinical director of the HRC and Community HealthTech, and I've also sat on the I4I funding, one of the I4I funding committees since 2020. And this is a particular area where I think it's very difficult to do it well, but doing it well really helps the committee to see why you should be funded compared to other people. So there's two areas that I'm going to focus on in this brief section. One is how one is trying to how you tell the committees that your product is different from other existing technology and convince them that it's really going to deliver a substantial improvement in patient care. The second thing that committees want to know is what evidence is there that your product works in the way you say it does. So effectively, is it low risk enough? These committees do fund into risk. They want to pick up risky products that could work, but is it low risk enough to make it worth investing in to develop it further? So the first way that you can do this is by outlining really clearly what the competing technologies might be for your new innovation. The committee really wants to know what will distinguish your product from technologies which are currently on the market or technologies which are advancing toward market. And to do this convincingly, it's quite helpful if you tell us how you've looked or how you know. So we've scanned using this or that database, or we're aware through this or this method of the competitors in this space. And be as comprehensive as you can. The composition of these funding committees is really broad, and there is often the case that there are technologies that the committee will be aware of. And if they're not mentioned in your bid, then that is worrisome. So be as broad as you can in your search, but also as broad as you can in considering the types of technologies that could be competing. So if you have, for example, a gait analysis based technology for a movement disorder, that could be one way to diagnose and monitor that disorder, but also brain scans could be used or blood biomarkers could be used. You'll need to cover all of those different types of technologies as competitors, not just the other types of gait technologies, which might work in the same kind of way that yours do. And having a sense of what they are then allows you to take the next step, which is to try and highlight what your new innovation offers that is different. And one of the more successful ways to do this is to do a sort of competitor figure in your applications. So then moving on to the second question, which is how do you provide a good proof of concept for your innovation? Well, clearly the nature of the evidence that's going to be required is going to be really variable because there's such a variability in the kinds of health tech that come through to these types of funding programs. But really what we need to see is the evidence that meets the use case that you're aiming for and what you want to achieve by the end of the grant. So there are many different starting points for this. You could be coming with some in vitro evidence or some animal evidence that then says that this technology could work in humans, and what you want to do next would be the first in man study. You could be coming with some early accuracy data of your new test in a design which takes multiple groups. So that means a design where you've only looked at totally healthy people and people who've definitely got your condition. And then you need to take the next step from there. You could be bringing in some technology that you know works for one population, but you have some evidence now that it could pivot into a different use case. And that initial evidence allows you to make the case for expanding your technology's remit through one of these funding schemes. Or you could have a good evidence of performance of your technology. You think it really does work, but now you need to put it into a context and develop the evidence for policy or commissioners that it can be clinically effective and cost effective in real world care. So many different starting points, and depending on where you're starting, and there are others from this list as well, it's about presenting the right kind of evidence for the committee to see that is reasonable and a reasonable place for you to start. So one way to think about this, and this is more used for diagnostics, is the idea of a diagnostic evidence cycle. So it's quite an old publication now, but Horvath and colleagues published this concept of how technology moves through a cycle of evidence and development, usually starting at the top with, if you're an analytical product, how you meet the analytical standards in a very controlled laboratory setting. If you were a device, how you perform on a phantom or how you perform meeting industry standards, then moving through how it would work in clinical performance, then how effective it is at achieving the aim that you want to improve, the health outcome that you want to improve, how cost effective it is providing value for money, by achieving that health outcome, and then broader consequences, which might include behavioural change, qualitative impacts, improvements in, improvements in staff retention, etc., which might come from the introduction of this new technology. It's useful to maybe think about this as one way in which you could sort of frame where your technology fits and where you're starting from for your application. Clearly, there's something here about who the evidence is generated on, in what setting, where it was generated, and that is important. Because we know that the incidence, particularly for diagnostics, we know that the incidence of a cancer might be very different in different countries. So we sort of want to know how close was this evidence to evidence that could improve NHS care. There's also mention here about the types of measures we use to describe the performance, the sensitivity, specificity, and also mention of the methodology that was used to achieve that results, the fact that it was blinded, the types of patients, the nature of the patients. These are the different things that I think I've added in here. So firstly, I've mentioned the study design. So this is a prospective diagnostic accuracy study. And that's really important because if you come in, there are various different ways in which you can compare a healthy group to another group. Prospective diagnostic accuracy would probably be considered the highest form of evidence you can provide. So if that's what you've done, you should definitely be telling the committee because they'll take account of that. So then I've also mentioned the location, and I've told you what kind of service. If this cancer biomarker is meant to work in primary care, where I work as a GP, I probably want to know at some point that it works on patients with undifferentiated symptoms like the ones I see, rather than just the ones who I've already spotted who've got bladder cancer and sent it to urology. But this is okay for early proof of concept data. I just want to know, I want to know either way. And then we also, next click, we want to know how many patients have been included. And that's obviously key because it affects how confident we can be in the performance of the test. Now, I've made up these performance characteristics, and actually they're a bit tight for a study with 60 patients. They'll probably be much wider than that. We'd have much less confidence in our findings. But I want to know how many patients came in so I can assess the diagnostic statistics in context of that. Then I want to know what kind of cancer they had. And here, stage 4 is the kind of metastatic cancer. So definitely, absolutely, definitely bladder cancer. And who I compared them with. I need to know what the reference standard was used. So what the methods were in a bit more detail. So I know that this was a good quality study. How do we know these people have bladder cancer? They'd had a biopsy, they'd had a scan. If it's a difficult thing to have a diagnosis of, what did you do to get as close to that as possible? Also, a bit about the quality of the methods. We'd want someone to be looking at your biomarker results who didn't know what they had, because it's very easy for humans to affect results even if they don't plan to. And then where you have got results, try to give as much detail as possible and give that detail in a way that would be considered academically or scientifically accurate. So accuracy, people a lot of the time in our line of work will say, well, it's this accurate, but what does that actually mean? That's not a sort of scientifically recognised term. So sensitivity or specificity or positive predictive value or likelihood ratios, something which kind of fits with the sort of academic understanding. And our service and the RSS can both help you with how you frame this evidence, because often it's the case that it exists. It's just that it's not written in a way that a committee can understand. So just to reiterate, give as much detailed evidence as you can, and then really justify why the project you're proposing in the grant forms the next required steps towards patient outcomes being improved. So explain where you're starting from, explain what you need to do next and why you'll end up at this, where you'll end up at the end of the project, and then articulate what the realistic next steps are and how you will build towards these in the grant, which kind of links to where you're starting from in terms of your proof of concept. So I was just going to go through the examples that I gave earlier. So, for example, if you present in vitro and animal evidence, then you might be saying, okay, well, the next step here would be that we need to take this evidence that we've shown works in a porcine heart valve into first in human studies or into cadaver studies, and that's what we want to do with the funding. If you've got early diagnostic accuracy data, you might want to then be going for a larger study, which is appropriately powered in a design which is prospective, so taking all comers rather than taking two groups you definitely know do and don't have a condition. expanding or addition of a new use case is an interesting one. I think this committee at least used to see quite a lot of this, and sometimes the evidence that the new use case could be supported with the existing technology is quite scanty because you haven't had that product for very long and you haven't got any good evidence, and that is more tricky, and I think might need sort of smaller amounts of funding to do some pilot work, to do some acceptability work, and drawing on evidence from other sources. And then if you've got a product with good evidence and you want a big clinical cost effectiveness piece of work in real life settings, that is also funded through the I4I scheme and is a really important step to then turn around to NICE and say, look, this really should be implemented.